Hello, my dear students. Welcome to my today's video lecture or online class on RAPD. RAPD means Random Amplified Polymorphic DNA. In the last class, you have learnt about RFLP, and this is another technique which is available, which is in use right now, especially in the field of plant taxonomy. This RAPD is also called arbitrarily primed PCR, ATPCR. I'll let you know why it is called so little later. I think I'll skip this slide. By now you know who I am and why my mobile number and mail ID are there on this slide. And you can see one more picture which I am going to explain it a little later, that gives the schematic representation of RAPD. Okay, that's the reason why I have highlighted it. Now, normally in PCR, what I have taught you in my previous lectures, the main objective is to generate defined fragments of DNA from highly specific primers. That's what we did in PCR. But what are we going to do in RAPD? See, in RAPD, short oligonucleotide primers are arbitrarily selected to amplify a set of DNA fragments randomly distributed throughout the genome. Right now, you just remember this. When I explain the technique with the schematic representation, you will understand it better. So, here you make use of oligonucleotide primers, which are arbitrarily selected to amplify a set of DNA fragments randomly distributed throughout the genome. Hence, RAPD is also called arbitrarily primed PCR. This is why it is called APPCR. Okay. But overall happening in RAPD, that is procedure, is comparable to the general technique of PCR. This method involves the use of a single primer at low stringency. Okay, let's see what exactly happens. Now, I told you the process is almost similar to PCR, but what are the differences or what are the comparisons? This slide will tell you the components of a PCR reaction and the components of an RAPD reaction. See, in both the cases, the buffer contains magnesium ions. But in RAPD, normally you give higher concentrations of magnesium ions. Why? For lowering annealing stringency. Okay? For lowering annealing stringency. So in RAPD, you give higher concentrations of magnesium ions compared to PCR. What is the template used? In both the cases, it is DNA. In PCR, as you have already learnt, two primers that flank the fragment of DNA to be amplified is seen. You make use of two primers which flank the region of the DNA to be amplified. Please note, but in RAPD, you make use of one short primer, normally 10 bases length, and not known to anneal to any specific part of template DNA, right? Randomly it goes and attaches to different regions of genome. This is the major difference between a PCR reaction and a RAPD reaction. Of course, in both you have to give DNTPs, that is ATP, GTP, CTP and TTP for furtherance of amplification. Then in both cases you make use of Taq polymerase that is thermostable polymerase, DNA polymerase, or any other thermally stable DNA polymerase. So if you are asked to write, compare and contrast a PCR reaction and an RFPD reaction, these are the points which you are going to highlight. Then, what is the principle of RFPD? What exactly is done? See, in the diagram you can see the DNA template, and that you are going to subject it to RFPD. You are going to put the primers randomly, right? And then the PCR reaction will take place. So you will get RFPD PCR products. You can see the two bands, two amplified DNA pieces, and that you can always separate out on gel electrophoresis, and you will get the banding pattern as shown in the diagram. So detection of the size of the amplified fragments is done, and therefore you can always compare. This is the basic principle of RFPD. Okay. So a short oligonucleotide primer of usually 9 to 10 base pair binds to many sites in the genome, and the DNA fragments are amplified from them. Then, the stringency of primer binding can be increased after a few PCR cycles, but initially in RFPD you have to keep stringency little lower only, and that is done by keeping a higher concentration of magnesium ions, right? Then when this happens, amplification of best mismatches will take place. That's the reason. Then RFPD can be carefully designed so that it finally yields genome specific band patterns that are useful for comparative analysis. This is possible because genomic DNA from two different individuals often produces different patterns by RFPD. I'll show you the diagram little later. You will understand this point better. Thus, a particular DNA fragment may be generated for one individual and same may not be generated for the other. And this represents DNA polymorphism, and that can be used as a genetic marker. So this is the platinum sentence, okay, in your answer. Therefore, I have highlighted. I'll repeat. A particular DNA fragment may be generated for one individual and the same may not be so in another individual, and therefore there occurs DNA polymorphism which can be used as a genetic marker. So this is the schematic representation highlighting the different steps. See, here we have two samples, sample A and sample B. We have isolated the genome or the DNA. What you are going to do is, you will give them DNA polymerase. Of course, by now you know it is Taq polymerase or any other thermostable suitable polymerase. And you will give short universal primers. Please note. Okay, you don't give two primers which anneal to the flanking regions of the DNA on either side. What you do in PCR? No, not at all. You will give short universal primers, oligonucleotides of 9 to 10 base pairs length, and you will conduct the PCR. You can see the figure A will show you the PCR reaction for sample A DNA, and figure B will show you the PCR reaction for sample B DNA. Then once the amplification is through, you will put them to electrophoresis, and you can see the banding patterns developed for sample A and for sample B, and definitely there is polymorphism. Okay, this is how you can make use of RAPD as a marker. Okay, in this scheme, the dotted lines will tell you that they are the primers, and the thick dark lines are the amplified fragments, and arrow mark will show you the direction of amplification. This is what is exactly done in RAPD. It is widely used by molecular biologists for genetic identification of plant species. So this has a wide application in molecular plant taxonomy. Okay. For this purpose, what they do is combinations of nucleotides. Most of them are oligonucleotide primers, as I have already told you. They have been designed and they are readily available commercially. Okay. Simply you can go buy the primers of your choice and do RAPD. As each random primer anneals to a different region of DNA, many different regions of loci on DNA can be identified. Okay. And therefore, RAPD is useful for the construction of genetic maps, and it also serves as a method for genomic fingerprinting. So, the two major applications of RAPD are number one, construction of genetic maps, and number two, they serve as method for genomic fingerprinting. With all this RAPD, though much better than RFLP, has its own limitations. The main problem is associated with reproducibility. Okay. It is often difficult to obtain similar levels of primer binding in different experiments, and therefore difficult to correlate the results obtained by different research groups on RAPD. This is the major limitation of RAPD. This slide will just compare the RAPD and RFLP techniques. Let us see one by one. I'll just zoom it. In RAPD, it is basically a PCR based technique for identifying genetic variation. But RFLP is a molecular method of genetic analysis which allows individuals to be identified based on unique patterns of restriction enzyme cutting in specific regions of DNA. So this is PCR based method and this is restriction digestion based method. Then RAPD involves amplification of genetic markers in a genome by using random primers, whereas RFLP involves restriction digestion of the genomic DNA by making use of restriction endonucleases. For RAPD, small quantity of DNA, 10 to 15 nanogram is enough for the analysis. On the other hand, RFLP requires comparatively large quantities of DNA, 2 to 10 micrograms. Here it is nanogram, here it is micrograms. Then RAPD uses random primers universal for any species as they are readily available commercially, whereas RFLP uses different species specific probes. Then RAPD has comparatively fewer steps and therefore it is a rapid process, whereas RFLP has more steps and therefore it is a slow process. RAPD is less reliable, whereas RFLP is more reliable. This is a difference. Then RAPD can detect 1 to 10 loci, whereas RFLP can detect only 1 to 3 loci. Then RAPD is not able to detect allelic variants. Allelic variants cannot be detected by RAPD, whereas RFLP can detect allelic variants. So these are the differences and comparisons between RAPD and RFLP. Okay. My dear students, thank you for your patient listening. Any questions, you can contact me either over my cell phone or through mail ID. Thank you for your patient listening. Stay blessed.